least wait another minute or so. Well, I'll just say a few comments to begin with. One is that it's been so long since I found out about this Nobel Prize that this presentation's changed a lot. In fact, one of the problems an hour ago was that I was looking at where it's really set up for the first version of this talk. So we're on cue here for what at least the most recent one. So let's just start. People show up. That's fine. Well, I always like to begin with a little perspective, and this is by Michael Shermer. Some of you may have seen this, but I like this because we all kind of think in narrow little holes. And I like this. What science tells us is that we are but one of hundreds of millions of species that evolved over the course of 3.5 billion years on a tiny planet among several orbiting an ordinary star, itself one of possibly hundreds of billions of solar systems in an ordinary galaxy, itself located in a cluster of galaxies not so different from trillions of other galaxies in an expanding universe that is very possibly only one of a near infinite number of universes. Wow, that's a lot of perspective. And we're going to return to that a little bit. And so I like this next slide because this is Leiden, or at least part of Leiden. And some of you can look at that and tell me the time of day. And you can kind of figure that out. You look for the shadows, right? And where is east? East is over there on the right, west over here, and you're all seated south. And looking up, it's a lovely weather. There's no wind. It's autumn, but not many leaves have fallen. So it's probably in October, something like that. And so it's actually a beautiful place, and the so-called Oxford of the Netherlands. But one of the things that really struck me that's so different than the stuff we hang up around Nobel sometimes in prominent places is they put murals up. And many of the murals, if not all of them, most of them anyway, are really equations, physicists. And this is the equation for general relativity. And so I'll just point out a little bit here, because the graphic is up above, but here on the right-hand side is really mass, defines mass, and on the left, at least on the left-hand side here, describes the curvature of space-time. So there it is, one line. Many physicists look at this and consider this absolutely beautiful, because in so few symbols, it describes the universe. And it's really quite extraordinary. And the illustration of how mass can actually bend light. In this little picture, there's the sun, there's Earth, and light from behind of the sun that you wouldn't ordinarily see is bent around it. And that's the lensing effect, which they use to sometimes see whole galaxies or several galaxies hidden between or behind interim galaxies, kind of blocking the way. So it's like a lens, and something that Einstein actually predicted, and it's a general tool these days in astronomy. So let's just move along. But I love these murals, and I think there are around 20 of them in Leiden, various places. And the other thing about this, he's not paying any attention to it at all. In fact, he looks a little cold. And so that too is real. And so well, one of the things I added in this is, because it is relevant to what we're going to be talking about, it doesn't seem obvious when we're talking about the immune system, but this is very important because biology is really moving very rapidly. And so let's just go the fertilized ovum, the single cell, it begins to divide and differentiate and specialize. And it becomes hundreds, if not thousands, of different, very specific cells, but all with the same DNA. How does that work? Well, it works because there's a system for silencing certain genes, activating others, all in a very exquisite order. And so that's differentiation. But you can reverse that process. That was really a breakthrough back in, say, the 1980s and 90s. I'll show you some of the foundational work here. But you can reverse that. So a very complex, mature cell can be re-engineered back to its more primitive or early stage, and then redirected to become a brain, to become part of a lung, to become something else. So that these are really very powerful tools. And what I'm going to talk about are brain organized. You can redirect these cells to become some small version of a brain. And what you may not know about is that artificial bases have been created, so we've been stuck with the 4 or 5, with the 4 for DNA, but you could actually substitute artificial bases. In fact, you can create DNA from scratch, and it will function like DNA. And the ominous part about this is that you could weaponize this kind of thing and to create very unique organisms that no human, no animal has ever been exposed to before, with no fix. And it's easy to do that. You don't need a huge lab to kind of create this kind of thing. So that's maybe not in our future, looking around the room, but maybe in our grandchildren's future. And so to the two men that created this revolution in reversing differentiation to a simpler form and then creating a whole animal from a single mature cell. Both of them did this. So John Gurdon from actually Oxford in the beginning of his career, but mostly at Cambridge, and Yamanaka in Japan. These are the ages which they got their Nobel Prize. And John died this year at 92. He's an exemplar for this kind of thing. Teachers can really screw up students. And here's a report from a biologist that taught John Gurdon. I believe he has ideas about becoming a scientist. Well, I don't have to read the stuff, but you see. And that delayed him getting into Oxford by 2 years. Within 3 years of getting into Oxford, 3 to 4, he had already done at least the initial parts of his experiment. So he revolutionized biology by showing that he could take a single nucleus, say from a mature intestinal tract, and insert that into a denucleated cell, and that would go on to create a tadpole and a frog. So there's that whole business of de-differentiation and then reversing that to a mature animal. That's really incredible. Now, were there a lot of failures? Of course, when you try to destroy a nucleus in a cell, probably many of your cells die. Maybe only 5% success rate. But anyway, so here he was, really quite extraordinary experiment. And then, well, we saw that. So now, and this is a kind of the cartoon from the Nobel Prize thing, showing that zapping the nucleus here and using a mature cell and substituting that into this one, where he destroyed the embryonic cell here, and it went on to create a tadpole and then the full frog. Isn't that incredible? It really is. And no one even thought about doing that kind of stuff before, say, the late 1980s. It's incredible. And here's Yamanaka from Japan, and what he did, he showed that there are transcription factors are factors that can influence whether a gene is active or not. And there are a lot of them. And he found that he only required 4 of them to convert a cultured adult mouse fibroblast and turn it into a pluripotent cell, meaning capable of creating the adult. So that's quite extraordinary, because he started with fibroblasts, a highly differentiated cell, and turned it around into a whole organism. It's incredible. And just going a little further on that, and it's also a way of looking at, say, what's actually going on in certain diseases. Well, you use the same process. You could take cells from, say, meat, might be fibroblasts in meat or muscle fibers, de-differentiate them to this pluripotent cell capable of becoming any cell, and then redirect them to becoming, in some cases, nerve cells, heart cells, muscle cells in the heart, or liver. And then you can assess or study those cells to see what's actually happening in the disease. I mean, it's a beautiful way of doing things. Again, no one's thinking this way at all until maybe the last 30 years. Let's see. Now, organoids. So we're talking about these stem cells that you can create. The lingo is induced pluripotent stem cells, or IPS cells. And they can be coaxed into becoming skin, any organ in the body, any system in the body, any type of cell. And the interesting thing that I've underlined it here, it may be difficult to culture these cells, but the cells themselves don't need any guidance. We just let the cells do what they want to do, and they will do it to become whatever cells you want. Isn't that extraordinary? It doesn't require any outside messaging from us. In a sense, they're self-organizing. You kind of set them together, and in a sense, they cook dessert or whatever it is without any intervention. And so it's possible to create mini versions of the brain. So we're back here. And they're not very big, kind of smaller than maybe half of my fingertip. They survive, some of them 5 years, 7 years. They can't grow beyond a certain size because they don't have any blood supply. And so they'll differentiate so far. But look, this is extraordinary. This kind of blew my mind, because the different colors here, that these cells have been stained, if you like, or marked, if you like, by for certain genes. And what's really extraordinary, that these cells started out, this would be the lateral ventricle here, and they divided and multiplied and became more and more complex and differentiated from one another. And they follow chemical signals from the cells that have gone this far into becoming 6-layer cortex here. So all of this is a marvelously scripted, genetically speaking, and orchestrated system for creating, in this case, 6-layered cortex, which looks like the equivalent in a human embryo at about the same age. And if you're looking for developmental diseases and trying to understand why certain kids are unfortunate enough to get this or that disorder, this is a great model for doing that. It's also a way for differentiating, say, between a Neanderthal brain and a human brain. It turns out that one amino acid difference in one protein defines the difference between a Neanderthal brain and a human brain. Isn't that extraordinary? But this is amazing because, and so I wrote this little piece here, Einstein watching creation at work. So just to reread this miracle in a way, stem cells bordering the rim of the ventricle differentiate into specialized cells, and they migrate to their destinations in the emerging neocortex. And they're guided by the cells that operate like ladders or scaffolding cells, directing them along the way or in response to these chemical signals, kind of drawing them on. But to watch the unfolding sequence is to learn how nature creates what is, in this instance, a part of the brain which evolved over several hundreds of millions of years into a highly sophisticated organ capable of understanding the universe we live in and how life evolved. It's really quite extraordinary, isn't it? It's another bit of perspective. And this is me imagining Einstein. He didn't know much biology, but perhaps as Einstein playfully imagined, the universe and everything in it, including the tiny planet on which we live, and the many millions of creatures which evolved over several billion years, point to a master, creator, and director for the whole shebang, and much, much more. I think it's that big, or bigger, the real story. And so it's certainly, and these days it's even more stunning because they're now going about assembling a whole brain. Because if you could create the neocortex, well then you also have to create the basal ganglia, you have to create the different lobes in the brain, the cerebellum, the brainstem, and that kind of thing. And they can all those parts can be created, and they can actually be, if you like, stitched together. And they actually talk to one another. And cells will send processes into the other parts of this brain. So will they ever be able to create the whole brain? I hope not in a way, but technically it clearly is possible to do this because the tools are there to do it. And so this is the lab in Harvard, and that keeps them going 5 to 7 years. And well, AI, where would we be without AI here? In several places, and this is quite new work, they can record electrical signals from the organoid, use AI to figure out what it's actually saying, what kind of sense it's making, and so that's down to step 5, use AI to analyze it. But the point about this is that I think the dawning realization of some groups, certainly in Google, or at Google, and some of them are very well trained biologists and neuroscientists as well as knowledgeable in AI. And I think the current thinking is that the human brain and AI share a lot in the way, a lot in common in the way that they learn. And you'll see this. Well, here we are. I've just sampled a little bit here. some people are very worried about these organoids, these brains, about whether they are sentient. Do they have feelings? And should they be protected in a way? Could brains and AI be combined? They've already been combined. And here's the real issue right down here, because the past, present, and future of intelligence. The explosion of artificial intelligence mirrors how human intelligence evolved. That's a really interesting claim, and I've read the review article, and it's very well done. There are some real similarities between the way our human brain learns and the way AI works, with the suggestion that they co-evolve. Well, if you think about it, AI was created by us, but it's taken on a certain identity all of its own. Well, here we go to something I was going to say as dull as immunology, but it really isn't. I think in my career, I never liked to hear immunologists talk, because slides were invariably covered with all kinds of different cells, with different kind of names on them, uninterpretable ones, and it was almost impossible for me to kind of follow whatever kind of lesson they were teaching. And the immune system is incredibly complex. It really is. And it does have a complexity about it that rivals the brain. And you'll see, and so when the Nobel Committee writes about this kind of stuff, you're looking at the really dumbed down version of the stuff, and you're partially going to get that from here on in. Anyways, there are the three, Mary Brunkow and Frederick Ramsdell. Those two work together a lot. And Segagachi, on the right-hand side, his work was independent of these two. And there are their backgrounds. Kyoto University, one of the Japanese universities like Oxford or Cambridge, that kind of caliber. And Mary Brunkow, Princeton University. But these two have kind of worked in California and kind of the West Coast, and their colleagues, and they're only a year apart. There she is. This is 4 a.m. And she found out that she's won the Nobel Prize. Now think about that. I mean, this is 2025. The work that she did that won the award was back in the 1990s. So, a long time. I mean, it's not that she hasn't been busy since, but the work that generated the Nobel Prize was done a long time ago. And there he is. This has to be the West Coast, maybe Northern California, or maybe, I don't know, it's autumn, right? And see the kind of the yellow there, and there he is. There's his water, a foam, so he can, in emergencies, if something happens. I didn't see any bear spray, and it's warm enough for him to be lightly dressed. He's a very interesting person. They were a very good team, the two of them. So, well, what about the immune system? And this is about as simple as it gets. It includes the bone marrow. Why the bone marrow? Because the bone marrow is the source of all of the cells in the immune system. That's where the stem cells for all the complex cells in the immune system come from. And broadly, they're B cells. B cells are ones that create antibodies. That's their product. And T cells, and we're going to be talking about T cells today. And the thymus gland, at our age, looking around me here, It's probably fairly vestigial, hard to find at this point, but quite robust in, say, the first 10, 20 years. And it's maybe one of the reasons why autoimmune diseases are less devastating in older age, because we're not as prone to autoimmune responses. So the thymus gland, and of course the whole system of lymph nodes and the network of lymph channels. And one thing that's only become apparent, say, in the last 5 to 10 years, is the gastrointestinal system has an immense contribution to the immune system. And just imagine all the stuff we eat that goes through the alimentary tract, and it's analyzed, and in a way it's first contact. And and the spleen, I always think that people can live without spleens. But anyway, so there's the symptom. Some of this I think I've already said. The T cells, these are a form of lymphocyte or white cell here, and some of them are given names here. Some of them are like presenter cells. They introduce a foreign agent or antigen to cells to see what those cells are going to do. So they kind of introduce. That's why they call them presenter T cells. Helper T cells that send signals that direct other immune cells to fight infections. And there are the killer cells, cytotoxic cells. So if a cell is targeted, these cells will destroy it. And what we're talking about today are regulatory cells, T-reg. So this is what the Nobel is all about, T-reg down here. So we're not talking about all the rest of it. We're not much today. If you think about it, when you're in utero and or just after birth, you haven't been in contact except through your mother and bit through the placenta, but you haven't been as exposed to the extraordinary diversity of proteins in the environment that could provoke a reaction. It could be an organism, a living organism, or just a protein that is foreign and provokes an adverse response. So how does that work? I mean, do we just kind of take all comers in this and kind of learn as you go along? Well, no, that's not how it works. What happens in the thymus gland is that there are multiple genes and parts of genes that combine in different combinations, but they produce an extraordinary diversity in the proteins that are on the surface of every cell in the immune system. And those cells are like identity tags, or Rita or Bill or Jane or whatever. So those proteins are very specific. And there are thousands and thousands and thousands of them. And so in a way, the body has a built-in defense system already for recognizing pathogens. It doesn't have to be acquired. And it says this, we have an extraordinary capacity to recognize antigen proteins, which we have never seen before. And look at this, almost unbelievable number, 10 to the 15th. That's 15 zeros after. That's extraordinary diversity in different proteins. And that's why I say this system is really, really complex. It's so complex that it really leads to the obvious question. If you've got that much diversity, how do you, maybe some of those proteins might provoke a response, a nasty one, maybe an autoimmune response, And so how do you get around that? Well, again, that's what this Nobel Prize is all about. So a very complex system. Now, I took this little model for me, one of the articles I had in the paper about the immune system, where I kind of condensed it for me so that I could understand it. So it's a little like the social system that we all have for sorting out who's in, who's out. And we don't destroy people, but that system does destroy outsiders such as bacteria and viruses, but sometimes, by mistake, attacks kin, own cells. That's what it does. And cell identity is expressed in the form of, I mentioned, those complex proteins, like little rods or sticks sticking out of the cell membrane that provide the identity for each cell. And now the thymus gland makes a big contribution here, the so-called central arm of the immune system, which is not, again, part of this Nobel Prize, but it is half of the immune system. The thymus gland, I think the next thing will actually show us this. Sorry, go back. Yeah, here we are. So there's the thymus gland. It's quite large in small kids. Those thymus cells on their surface express on the surface a variety of proteins that are self proteins, meaning they were created by that child. And they're very specific to it. And other T cells circulating through the thymus gland are really tested. And if they have a locking, like a lock and key, if they have something that actually connects with a self protein on the surface, they're destroyed to avoid an autoimmune response. So it's really kind of pre-filtering out these cells that might provoke an autoimmune response, but it's not perfect. I mean, some of those cells actually escape, and that's where this Nobel comes in. Now here's Sakaguchi in the 1980s and 90s, shows you how far back it goes. Well, just look at this, how simple, now it's fairly simple. Discovered that injecting T cells from normal mice into mice whose thymus gland had been removed a few days after birth, say the third day, protected the mice from an autoimmune attack provoked by thymectomy, meaning if you take the thymus gland out, mice will develop a really violent autoimmune response that destroys the mice. But if you inject adult T cells into those animals, they don't get the autoimmune response. And that was his first contribution, identifying suppressor T cells, or T-regs here. And if I include another decade of his work, he realized or found out that, well, it wasn't all the T cells that he injected, it was a special group that expressed this protein, CD25, on the surface. And that's where, so it was a very specific group of cells that conferred this ability to suppress an autoimmune response. Oh, here, I'm slipping out of here. So, others supported this. But now the story gets a little more interesting. Well, here's the kind of idiot cartoon here. Newborn mouse here. They take out the thymus gland in 3 days or 5 days, somewhere in there, but very early. And if you don't do anything at all, the animal develops this autoimmune response, becomes sick, and dies. But if you inject adult T cells into it, they suppresses the autoimmune response, and they're healthy. But going further, he showed, well, okay, yes, we injected T cells into these mice, but which T cells? And it turned out that a very special protein on the surface of those T cells was the missing link. So T cells that expressed that CD25 on their surface, they were the ones that conferred that specific protection against an autoimmune response. So that was very critical work. Now, there are other experimental models, and one of them includes the scurfy male mice, and they're born with scaly, flaky skin, very large lymph glands and spleen, and they only live a few days because it's a very bad autoimmune disease. Well, it turns out it's genetically transmitted, and that the gene is a mutation on the X chromosome. So females have XX, males YX. If the X in a male happens to be the abnormal one, he is going to develop the autoimmune disease. If he's fortunate enough to inherit the X, he won't. So, they were very interested, okay, what's the gene that's kind of responsible for this? Now, you know, here we are in the 1925 going on 26, when it only takes 1 or 2 days to survey in considerable detail the whole genome. In those days, it took years to do the same kind of thing. It's an incredible amount of work back then. So, but they were very interested in kind of writing it down, and so we'll see a little bit of that. How much of a challenge was it? Well, the string of DNA, 170 million base pairs. Where in all of that is the abnormal gene? Well, and then they kind of looked at it and they figured out, well, it probably is in the middle of the X chromosome. Well, that narrowed it down to 500,000, but that's a real problem. And then they finally, we're talking 10 years from here to here, down to 20 genes, and finally the gene. And so, and it was what, the so-called FOX genes that control transcription, meaning whether a gene could be active or silenced or not. And so it's a mutation in that gene. And here's a little cartoon that goes along with it. There's the string of those are the kind of the 20 genes, and one of them was the FOXP3 gene responsible for the mutation and the disease. Well, you know, to solve this kind of mystery these days would be a piece of cake. But back then, in the 1990s, this took 10 to 15 years, if you include the three of them. And, but they did show the abnormal gene for the scruffy mice, but they also looked for, are there human autoimmune disorders with the same genetic abnormality? Yes, there is. So that was a big score to find that. And then the Japanese group showed that that same FOXP3 gene was responsible for the effects of the regulatory T cells that control autoimmunity. That's a beautiful story. It's a very solid story. It took 15 years to solve it, and that's what the Nobel was really all about. It's great detective work. You know, it takes an extraordinary patience and persistence. I mean, just running down, what was it, 170 million possibilities down to one. That's extraordinary. Now, I put up this slide because immunologists can make any discussion of immunological disease and processes extremely complex. This is the next level up. I could make this much worse. So, which we're not going to go to. So there it is. So wrapping up. So there is a peripheral component to the immune system, and these T regulatory cells are vital to controlling or preventing autoimmune disease. And getting to autoimmune disease, that affects a lot of organs, and on the neurological side, and I certainly saw in my career, myasthenia gravis and multiple sclerosis. Multiple sclerosis primarily women in their late teens through early 20s, and can be devastating. And myasthenia gravis, very difficult to treat in younger people, but in our age group, much easier to treat. Remember I talked about the thymus gland atrophy earlier, so that's probably part of it. And rheumatoid arthritis, well, you know some of these autoimmune diseases, but here's MS, and very typical. You know, the lesions are around the ventricles here, and a lot of these lesions are clinically silent. They don't actually produce symptoms identifiably, surprisingly. Is that me or you?
That's you.
Oh. Well, I'm going to turn it off. I know what it's about. It's readable what this is about. So, yeah, sorry about that. That's my doctor trying to get hold of me. So anyway, now here's where the disability comes, because here's the spinal cord, and there are lesions right in the brainstem here, mid-cervical cord, and then all the way down in the low cervical cord, and so this person would have had a lot of difficulty walking and been off balance. But this person's relatively young, right? Because the vertebrae all look pretty normal, and the disc bases look normal. So, now this is a typical or kind of an average kind of course with MS or any autoimmune disease, meaning there are flare-ups from time to time, whether it affects your skin or whatever the organ is or tissue involved. There are relapses, and these are designated by these little pink bars along here, and progressive disability here, and even cognitive decline. And you can see the brain, the atrophy, the ventricles getting larger and larger and larger, and that probably accounts for the cognitive decline. So that's a pretty typical course for MS. This is rheumatoid arthritis, and you can see how destroyed. Look at the. It really affects the MP joints, these joints right here, mostly. And you can see how the fingers get off to the side. And you'll recognize this, Renoir, who lived to be 78, I think. And look at his fingers, just like the X-ray. And people had to stuff paintbrushes in his hand, kind of bandaged up for him to continue to paint, that kind of thing. So that autoimmune disease is very disabling for him. Which brings us to the treatment or management of autoimmune diseases. For most of my career, that involved corticosteroids, prednisone, something like that. A lot of side effects associated with that, particularly if somebody's committed to it for a long period of time, and can be pretty effective, but lots of cases where it's not. And this is true for for many of the treatments until recently, when targeting the immune system, very specific targets, have really, it's kind of like magic. And the goal here is to kind of, well, if the problem is in the immune system, can we rebalance the immune system? And one of it is, autologous means own. So if you take somebody's own bone marrow and stem cells and then manipulate them so that they can target the cells that are causing the trouble, then that's a very specific treatment without any kind of spread to other things. It's very specific to a given target. It's been used very effectively in cancer treatment because you're targeting cells, really parts of cells that are causing the malignancy. But now in autoimmune disease, they're really very effective. And you probably heard about CAR T cells. You probably know somebody who's been with the age group around here. Somebody's had CAR T cell therapy. What is CAR or C? It's chimeric, meaning the cells they've actually combined some of the genes that create these proteins that are so effective on the cell, but it's like having cousins together in the same cell. I don't know whether that's very clear to you or not, but that's how it works, and it's incredibly effective and targeted. That's the main thing, it's targeted, without destroying the rest of the immune system. It's already known that CAR T cell therapy works really well for B cell cancers and solid organ cancers, but in autoimmune diseases, it seems to be very effective and very specific. Listen, in this town, probably past the age of 70, the chance of somebody being on some monoclonal antibody treatment is very high because they've become very effective and targeted. And this CAR T cell therapy is even more so. So this is really a product of success with, and here's an example of it actually. Here's a kidney, a glomerulus in the kidney, untreated. These are deposits of a protein, IgG, and they're fluorescing here. After treatment, almost normal, and highly targeted. It's really quite extraordinary, and that's going to attract a Nobel for sure in the next 5 years or less. Clinical trials, well, one of the things, since we were talking about the peripheral arm of the immune system and these T regulatory cells, one of them is, well, what can we do with the T regulatory cells? Can we boost them in some way, their activity? Well, you can. You can boost them. You can take the person's own bone marrow and treat it in such a way that you enhance the T cell effectiveness, the T regulatory cell effectiveness, and then reinfuse it back into the patient. Very, very effective. So there's another thing, these T regulatory cells actually work against you in cancer, because some cancers kind of figure out that if they can suppress the body's immune response to the cancer, then it actually favors these T regulatory cells, right? It's counterintuitive. I know it sounds that way. So now aging and autoimmune disease, well, I've already made some comments about it. I can tell you at least one Nobel laureate is taking rapamycin now, and I can tell you a whole bunch of people are going to start taking these glucagon-like peptides, GLP-1, the ones primarily introduced to reduce weight, but now we're finding out it reduces the risk of stroke and heart attack, all kinds of other so good effects. But this whole business of aging, could humans live to 150? Well, I don't want to live to 150, but why not? And here's an interesting example that you may have seen, and it was in Nature, but it ended up in the New York Times. Here's a lady who, I think she died just in the last 2 or 3 years, lived to 117. She had been tracked and followed for a long time. And then she has 2 daughters. That tells you right away. They're in their 90s. So clearly genes are playing a part here. She exercised, ate a Mediterranean diet, 3 portions of yogurt per day, liked reading, and reads like all the things we should be doing. And she was lean, and she had a healthy gut microbiome, an immune system, no inflammatory factors, managed to survive COVID quite well, though none of the risky genes. And she was lucky enough to have a variant of a gene seen in some dogs, worms, and flies that are associated with extreme lifetimes. Leaving me to wonder why, with such a sterling health record, she died. Oops. And yeah, what happened? You seem to be doing everything right. And which brought me to this. This is from Alaska. So my daughter and her husband and my grandson have 2 cats, and they're very different because Mario's the one cat underneath, and Mario's about 11, lazy, eats. He would be content to eat and sleep. Enzo is quite different. Enzo is 3.5 now, loves to be outdoors in what in effect is wild country, and has managed to survive 3.5 years through the night and the day. He can scramble up anything that's vertical and get away, and beats up on Mario. So, but the article here was, even cats are going on these drugs, and dogs. And by the way, I brought up cats to address some of the balance in this community, because all I see are dogs. And as marvelous as dogs are, I'd like to see the odd cat. But I've only seen one person in the last several months actually walking a cat, and not on a leash. And cats, just to finish off with this, when did cats become domesticated? Fairly recently, maybe 3,000 years ago, 3,000 or 4,000 years ago. A big deal in Egypt, worshipped in Egypt. And whereas, for example, dogs, their history goes back 20,000 years plus. And so I don't know why cats were slower to do this, but they were. And that just shows you that the lineage of wild cats were spreading into Europe going back, let's say, 10,000 to 11,000 years. But the domesticated ones from largely Egypt, only that, say, 2,000 to 3,000 years. So it's kind of an interesting little thing. And one other thing was with Mr. T, or Presidential T, discouraging foreigners from coming to universities. Listen, if you look back over the Nobel Prizes, the bulk of them have been awarded to Americans, meaning people who have American citizenship. And the bulk of them are refugees. Either first, second, or third generation. And so you're really cutting yourself off to kind of, and she was from Hungary, and he was from Germany, originally the family. And that's the mRNA pair. I think, and this fellow from Jordan, who won the chemistry prize this year. He's from Jordan. And so just to show that, you know, I'm going to finish on that, but so some things, when you screw up on something as fundamental as, well, where did Americans come from? Where did Canadians come from anyway? My parents came from somewhere. Except for kind of Aboriginal peoples, we've all come from somewhere. And why would we even think about trying to do them? Anyway, it's appalling. So on that note, I'm going to finish. This is probably the most challenging I found for me, partly because of my aversion to the immune system, but also because just it's a very complex system. And you were hearing the very, very reduced version of that. And it's very hard sometimes to condense something. That's certainly what I found here. But I certainly credit them. But I'll say the marker for where we are now in studying the immune system is with the new therapies, like the CAR therapy. Not the stuff that we're looking at here. This is old stuff here, and this work was done in the late 80s and 90s. The same thing happened with the physics prize, with the kind of tunneling business, and if I look at what's happening to physics these days, they're way ahead past that. I think what the and it highlights how the Nobel nominating committee kind of works, because they're really looking for foundational work, but they want evidence that it worked, that the story turned out all right. They don't want to be embarrassed by endorsing something and then it doesn't work out that way. So there's an innate conservative streak, at least in the sciences. And I don't know what Valmai or David would say for their Nobel Prizes, but that's my sense of looking at it. I'm not discrediting the stuff. I think it's marvelous stuff, and at least at a level that I can personally understand. But today, I'd be right out of it. It's become very complex. So this rounds out the Nobel series this year. Next year, Steve.
Well, just a quick.
It's never quick with you, Steve.
You had mentioned cancer, and I'm glad
Yes.
you did, and you had mentioned all through this the immune system. For someone who has certain types of cancer, it's called immunotherapy now.
Yeah, immunotherapy.
Yeah, and I was wondering, that's all because of this kind of research that's gone on? I mean, they're doing wonderful things. I was told it's natural. It's not like chemo that is chemical. Immunotherapy, I believe I was told it's more of a natural thing. Now, do you follow that? Or should I do all
Worry
over?
when somebody mentions natural,
Yeah.
because there are lots of stuff out there on YouTube promising to be natural, but if you mean taking advantage of what we know about the immune system to craft a better therapy, yes, I think definitely.
Well, I'm speaking of, say, cancer cells in the bloodstream.
Yes.
And what they told me
Yeah.
is with immunotherapy, they boost the immune system so that the white blood cells
Right.
can attack the cancer cells better.
Yeah, that was the subject of the Nobel Prize about 3 years into this series.
Oh, I see.
And yes, it makes sense, because cancer cells have an identity card and it's different than the surrounding cells.
Mm-hmm.
So if you can craft a cell or a cell group to target that specific
Right.
not necessarily the cell, but the protein marker that identifies it as Steve or Bill, then it can be very effective.
It's leaps and bounds, I believe, now.
Yeah,
Leaps
yeah.
and bounds.
The problem is being sure that other cells in the body are lookalikes and might be targeted by the same therapy. But yes, I mean, the chemotherapy up to maybe 15 years ago was really chemical chemotherapy, right? Kind of nasty stuff. And these, as you put it, more natural ones, taking advantage of certain cells in the body, reprogramming them, so they'll target specific cells. That makes a lot of sense, absolutely sense, and I
Thank
hope
you.
that got across. Yeah, yes.
Yeah, I had probably a lot of groups on program 60 Minutes, November 30th. It was about genetic engineering. And
Yeah.
It was a high school competition. And it was a high school outside of Georgia, or outside of Atlanta, Lambert High School. And they had a team of 10. They were all Asian Americans and they formed the team. So they lost, because they were first generation Asian Americans, because of DEI, they lost their funding. But then they got it back, and they became one of the 14 American teams to go to this competition in Paris. So in Paris, so they were working on Lyme disease, and they identified a protein that was developed from the beginning of Lyme disease, so they were able to isolate it, and we used the CRISPR data to be able to identify the disease within a couple of days of catching the disease, and then they were starting to not only identify it, but to work on a solution. So they entered the competition, and they went to Paris, and they were looking to win, of course, but they became in the top 10. And it was kind of interesting. They were the only American team to qualify in the top 10. This was a high school competition. But the interesting thing is, one of the interesting things, 7 of the top prizes, so it's top 10 were Chinese.
Well, that doesn't surprise me at all.
No, but here's the interesting thing,
Yeah.
the most interesting thing. They got, I tuned into the internet and got the winning competition. So there were 10 people up there. And they explained there was something about interior household mold. And each of them spoke a bit about the presentation. They could all speak perfect English. The 10 of them. And they explained their project. They went one, and this would explain one part of their mold analysis, 2, 3, all the way up to 10. 10 people all spoke English. And I mean English, not English you couldn't understand.
As a group, they're very diligent, and unlike maybe some here, maybe more native work, and if you look at, I've noticed this in the last 10 years. If you look at really good science sources and look at, okay, who are the first authors? Because those are the graduate students, postdocs, and that kind of thing, and the leader of the whole thing is at the end. How many of those names, I don't know that they're Chinese, but they certainly read as Chinese. And there have been many in the United States, and they're not there as spies. They're there for the reason we went to Europe 50 years ago, because Europe was the place to go if you want to know anything about quantum physics or chemistry, that kind of thing. But and so, and I think there's a fair bit of collaborative work, but that's become more awkward with the current administration. And no, I'm not surprised about that at all. They now, this is just in the last 5 years, they now regularly rank in the top 10. I'm speaking now in science, and if you put all of the sciences, the hard sciences together, the majority are in the top 10. And if you look at the institutions, the majority are Chinese institutions. They've been very intentional about developing world-class institutions. And they also go after, I have a very good friend who was a graduate student working with me at one time, some time ago, and he's Chinese Canadian. And he's been, they've been after him, and not only after Ming, but after some of his Chinese students, Chinese Canadians and Chinese, who have collaborated with him. Again, you can't blame them, they're after talent, they're looking for talent. And why not? And, but with so much defunding now, the focus has shifted, right? That many Europeans and maybe not a few Americans and not Chinese Americans, are going to China. Boy, we never saw that arrow going that way. And so, yeah.
I was intrigued by your comment on organelles, neural
Oh, yes,
organelles,
yeah.
and little brains.
Little brains, yeah.
Our brains cost us about a watt or something like that to run.
They do.
AI costs orders of magnitude more than that. In fact, we're
They do.
worried that if they have any more clouds out there, we're gonna run out of energy. So I'm intrigued by the idea that someone in the United Kingdom already is using these neural organelles to program and perhaps substitute them for the iCloud out there and save our energy.
Kind of a green way of doing it.
It's an intriguing idea, and it might save us
Yeah.
eventually if we can get rid of silicone brains and use our carbon-based brains
Yeah.
instead.
ChatGPT, at least all of AI, is an energy hog.
Did any of your references in nature to using these organelles, these neural organelles, refer to that? Someone is trying to program those organelles,
Yes.
those little brains,
Yeah.
and figure out how to
Well, they are.
use them as computers.
Trying to see to what extent they can use them that way. And they're at the very earliest stages.
Very,
So
yeah.
when I look at something like that, I say, wow, we've seen this kind of exponential growth before. And it's not going to grow up like that in 5 years or 10 years. It'll be a lot better than that. So, and a lot more effective. So I think it's very interesting that people are thinking about hybrids of human organoids or something like that, and patched together with AI. And interestingly, that the fellow I mentioned to you earlier, the one with neuroscience background, but also in AI, his hypothesis, Google's one, since he heads the division, is that AI learns computationally, and that humans do too. Now you're going to ask me what's the definition of computational, but meaning in a reasoning way. If if not, it kind of, because that's how AI actually solves problems. And humans, he would claim that has been part of what has driven evolution. What's been the engine of evolution? How did we, how did we take advantage of our environment? How did we figure it out to our advantage? That's computational learning, in his view, and not so different than the way AI learns. And both of them have a flexibility for changing. It sounds very mechanical to say this, but they're the algorithms, the way they think. the rules that govern their thinking. We don't like to think that we're controlled that way, but we are. We are prisoners of the way we've been taught and of our biology at some level. So I think he's on to something. I think there's a common ground. I don't know how much common ground, but there is common ground between the two. And it's a fascinating way to think about evolution. Because no one's actually, evolution as a tool, what's actually happening, why do species evolve, hasn't changed all that much from Darwin's time. And talking about the tools, what's actually happening biologically in the organism, and how did this creature take advantage of that mutation, whatever. So I think it's an interesting way to go, and Google doesn't spend money foolishly. They've won 2 Nobel Prizes already last year, right, in physics and chemistry. Not, the name was never acknowledged, but the program was Google in both cases. And I see it creeping into a lot of analytic studies of using tools like Google originated and others. So I think that's the way it's going, and it's because it can solve problems that are tremendously complex. I mean, trying to solve part of our power is that we're a collective. We don't depend on worrying about things just on our own. We learn from the society, the people we know, and we find out stuff. But you couldn't run any of those telescopes without a team. And that's the point, I think. So we have a collective intelligence, and that's probably more important than our individual intelligence. Much as I like to focus on Einstein, but the Einsteins are rare now. Meaning a single individual with thought experiments that are really very original, that don't depend on a lot of other people, and depend on collaborative work. Yeah.
The difference I see is that for silicon, for AI, as it is now, they rely on a few things like electrons and bits, but nothing but electrons coming and going. Whereas we depend on those 10 to the 15 different molecules in our computing system. So if AI can do all it's doing with just electrons and things,
10 to the 15.
Can we do a whole lot more with this huge complex of interacting molecules? Doesn't that make us qualitatively different?
We are qualitatively different. Don't get me wrong. We're not walking computers. And
I'm just saying we're going to be smarter than that.
Yeah, but I do think that the nature of some of the problems that humans have taken on absolutely require groups, and the obvious ones are the telescopes, and even bodily physics these days. Schrodinger is a standalone person like him, or Heisenberg, or Bohr, or Einstein. Those first 3 decades of the last century, there's nothing like that around now. People really do depend on others, and it's a little discomforting. But as a species, I mean, roll back our story say, a million years, and we have a maximum of 1 million people, presumably worldwide, the genetic estimate, something like that. And they're living in so small scattered groups, and really scattered, of maybe 20 to 60. There's not going to be a lot of collective learning going on, or sharing with other groups. Sharing is a big deal, to travel 10 miles to meet somebody that you might not agree with, and not have a common interest with. So you can see that this evolution of the product of human intelligence, that's why it's a hockey stick phenomenon. It's really grown in the last 100 years, 150 years. I just look at, never mind the arts, I look at science and I look at that and I say, holy mackerel, this is an exponential curve up. And that's what it is, and it's because there are 8 billion of us now. There are many, many, many more physicists now than there were in Einstein's day. And that collective intellect, if you like, is what drives a lot of the stuff. And just imagine working at Google, where they kind of encourage you. They've got kind of separate kind of groups, but the groups talk to one another, they feed on one another, and so that collective intelligence is very, very productive. But it's beyond one person. I think Steven Weinberg was right. I've quoted him before, but the Nobel laureate in physics some years, he died, I think, 10 years ago. But he had 2 quotes that I really liked. One was that you want to learn something, teach it. I absolutely agree with that one. But the other thing was human intelligence has a limit. And we may be close to that individual limit now. And I hesitate to say that because where's the evidence? But I think part of the evidence is hidden in the fact that we do work collaboratively. And so much of the progress has come from the collective cooperation of humans. And that's what China is so good at, really, isn't it? Of putting people together in a place, supporting them with all the tools that they need, and encouraging them to travel abroad, and encouraging people who are abroad to come home, and so that collective intelligence really has an impact. I mean, look at where they were in the Korean War and where they are now. It's a huge difference. Okay, well, so on to next year. I was wrong this year. I thought I could predict it, but I lost it. I thought those diabetes drugs, the GLP, the thing would win, and it will. It'll happen in 2 or 3 years, but I got that one wrong. Yeah, one last.
One last one, thank you. Could you say something about the relationship between mind and body, and any causal or contributory effect that the mind has on psychosomatic or functional neurological disorders?
Well, I think they're intimately related.
Mm-hmm.
It's almost an abstraction to draw them out, and it's stating it that way is a disservice to. And I don't mean your way of looking at it, but when you draw it, when you make a distinction like that, it's an artificial one. I think they're the same. Listen, we're not aware of Jade and I talk about this all the time. We're not aware of 99.99% of the stuff that goes on in here, fortunately. And so establishing causality or relationship of this to that is a very difficult thing to do when you don't actually understand how the human brain is actually working. And that business I brought up with the gut, one of the things I didn't mention today was that there was a Nobel prize given 2 or 3 years ago for mechanical receptor, touch pressure figures, and it was very beautiful work. The same guy, Patapudia, he realizes that kind of almost 95% of the sensors in the body are in the inside. They're in their gut. They're in the organs that are kind of alignment. In other words, there's a whole lot of information that's taking place that we don't have a clue about what's going on. But the body uses it to communicate between organs and systems. And so is it any surprise then that we have trouble understanding what some people in prior days would have said psychosomatic symptoms? The implication being they were a little nuts. But I think a lot of those things have probably real biological origins, and it makes a lot of sense to begin to think that way, and that's where Patapudia is going. And I'll follow him. I think he's right. So most Nobel laureates kind of stop when they get the prize, and they do. They big travel things, and then everyone forgets them after a year or 6 months. But they're not continuing to be productive. He is. That's an unusual person. And they've also reached an age, but the people we're talking about today are in their kind of midlife or older. Hardly where you're going to expect novelty and energy to do stuff. That's people that don't look like us. no critique of us, but I mean, the truth is, that's true. Yeah. So I don't know if I've answered your question, but I think we're really far more complex than we recognize, and we use words and language sometimes to express things that we don't really have a clue about, or not much. And a lot of it has to do with the emotional side of the brain and feelings. We're better at math. Well, some are, not me. Okay, so we'll see. Where is it? So there's a series coming up in New York, but on AI. Yeah, so that'll be a good one.
Here.
Okay.